Obrigado, and bom dia. That makes it easy for the translator. I first want to thank the organizers for allowing me the opportunity to speak to you today. And I just want to clarify, my little box didn't work properly. I hope you understand I'm not with the Food and Drug Administration. At this point, I had worked for them for 20 years. So I'm not speaking certainly on behalf of the US government, but I am speaking to you on behalf of people who regulate. And what better way to start a forum such as this to talk about the subject which I know that all of you are very, very excited about, and that is regulation. That was a joke. But seriously, regulation is critical. But first, before I begin, I'd like to start with just a little audience participation, if I could, so I can have a better understanding of the group of people that are here. I see two views of regulation out there. One is this one, which is regulation prevents access and it prevents innovation from taking place. The other is that of protection of public health and protection of individual health. I want you to raise your hand. Do not be afraid. I need you to be honest, just so I have an understanding. How many people look at regulation as the one on the left, preventing access, preventing innovation? Oh, I'm surprised. How many would say it protects public health and protects individual health? Good, okay, I like my audience then. I would like you either way. And I've stopped. Here we go. So what I'm going to do today is to talk about how a regulatory body can keep up with a public health emergency and with advancing technology and still assure the safety and effectiveness of the product. Because after all, that's what regulatory agencies do. They are responsible for putting into the hands of clinicians, in the hands of users, in the hands of caregivers, products that do what they are supposed to do every time, give accurate results so you know what to expect and you can trust the results to help you in your clinical work. So the first subject I'm going to discuss here is regulation during a public health emergency. So the conventional approach to regulation is we have risk. As the risk goes up, every product has some risk associated with it. As the risk goes up, that means generally a regulator is going to require more information to understand whether the product is indeed safe and effective. The more information there is to review, to assure determine safety and effectiveness, the more time is required. But ultimately, what the goal of the regulator is to make sure that the benefits of the product outweigh the risks associated with the product. Okay. This is what's normally done. It's different in an emergency. So their course is very high risk. However, time is of the essence, such that time isn't just money, but time is lives. The longer it takes for a product to get out the door, the more people are going to be injured or may also die. So in that case, a regulator should be looking at less information and doing it more efficiently. So again, the conventional way of looking at this is look at the product to assure safety and effectiveness, but during an emergency, it's different. During an emergency, we're looking for a reasonable assurance of safety and effectiveness in the shortest possible time. It doesn't have to be perfect, but it needs to get out there as quickly as possible with some evidence that the product actually works. So, how do regulators do this? Well, there are two systems that have been developed. One from the FDA, the other from the World Health Organization. The FDA has a pathway called the Emergency Use Authorization, abbreviated as EUA, and WHO has the Emergency Use Assessment and Listing Program, or EUAL. I'm just going to describe these for you briefly. So, there are different elements here. To use these pathways, a public health emergency needs to be declared, for both pathways. Technical documentation is reviewed relating to safety and performance, but that's limited. It's much less than would be needed for a traditional approval. The FDA does not need to review documentation related to the manufacturer's quality management system, whereas WHO does. They are a bit more stringent. An independent laboratory evaluation of the product's performance is also not needed for FDA, whereas WHO rather does require some. In both cases, there is time-limited use. So until this authorization and marketing is permitted, this use is permitted for as long as the emergency has been declared to address the public health issues during the emergency. Here's an important piece that is a big difference, the review time frame. I'm sure all of you realize that it can take months, if not years, for a product to get out the door. For an emergency use authorization from FDA, the approval can take place within hours to days, and I've seen that happen in my time at the FDA. What's critical also is adverse event monitoring and reporting. So during the course of the use of the product during this emergency, then any adverse events which take place during the course of using the product are reported back to the regulatory agency to make sure that the product is actually working sufficiently and providing benefit rather than too much risk. Just to show you some of the accomplishments, there have been 19 EUAs which have been granted for Zika and 11 Ebolas. FDA also has, in the past, a number of other EUAs that have been granted. So there are 30 EUAs for Zika and Ebola combined by FDA, and 10 that were done by the World Health Organization. Again, WHO is a bit more stringent in its review. I also want you to notice the difference between Zika and Ebola. There are almost twice as many tests that have been approved, granted an EUA by FDA. One of the reasons for that probably is because of market. Zika was a threat to the US, and so more manufacturers stepped up because they saw more of a market. This is my own speculation, compared to Ebola, but it's still significant. The 11 Ebola tests were granted EUA. So, what are some of the challenges during a public health emergency? First of all, test developers typically don't have access to specimens needed for test development. They can't make a test if they don't have the specimens to show how well the test can actually perform. At the same time, incentives are needed for test development. The manufacturers need to know that they are going to be able to get something from this other than altruism. And so incentives are actually needed to move that process. There are also, in the case of Ebola, certainly, there was evidence of duplication of effort for clinical studies, for submissions and inspections, each country wanting, in some cases, wanting to do a complete clinical trial before deploying the test, which meant that precious time was lost. What I'm going to do in the next few slides is to describe for you a framework for regulating emergency use IVDs. This was actually published in the African Journal for Laboratory Medicine. I think it was within the past year, and you can read more about it in detail there. The evaluation of the IVD is typically done by the regulatory body. Regulation oversees that, but there are several other pieces that impact the process of getting an emergency use IVD out the door. And I'm going to talk about each of these pieces in just a bit of detail. So first, and again, these are issues that will affect the regulation and to move the product along such that it can eventually be available for emergency use. And here I'm talking about an outbreak as an example of emergency. So during when the outbreak first has to be acknowledged that it exists, and WHO would be notified, as well as appropriate global health authorities. Specimens will be collected from affected individuals, and investigations would be conducted to actually identify what the infectious agent is. And then that information would be shared amongst those who need to know, that will allow the next step, which is development of the IVD. For the development, the disease process needs to be identified, particularly markers of infection that are going to be used to detect the infectious agent after the signs and symptoms develop in a patient. Then the manufacturer will identify a candidate, in this case an RDT, which is a rapid diagnostic test, because these types of tests are best used in point-of-care settings. And then communicate a target product profile, what the test should ideally look like, what it should do, what its performance characteristics would be expected. Then the tests are developed by the manufacturer that would meet the target product profile, and specimens would be made available to the manufacturers for development. And now we come to the evaluation, which is really the true focus of the regulator after having gone through those first two steps. And there are two pathways that I'm outlining here. Pathway 1 refers to an unassessed RDT. The manufacturer is going directly to the country that's affected by the public health emergency. The country assesses the product, identifies and resolves any issues, and then accepts the product for outbreak use. Pathway 2 involves starting with either FDA or WHO. In either case, the public health emergency is identified, invoking the emergency situation. The FDA will grant an RDT, the test an EUA, or the WHO will grant an emergency use through its pathway. What I'm saying here is that it wouldn't be necessary for the country to waste time, waste energy, waste resources on doing additional evaluations, but rather rely on the FDA or WHO analysis and get that test out there as quickly as possible. The IVD will be implemented, so it will be distributed. Depending upon whether specimens are available, then users could be trained using proficiency panels. If they are available, evaluation panels can be used to confirm the results, although this is not typically the case during an emergency, and a system for reporting test results to the Ministry of Health and other necessary public health authorities would need to be done. And finally, monitoring the IVD. This is critical. Adverse event reporting. There needs to be a system for the test manufacturer. This is required. So the manufacturer can acquire information on whether the test is performing as expected or worse if it's not performing and people, uninfected people, for example, are being put through are not being treated or identified. Each country should actually generate its own monitoring plan for emergency use. So the first part of the talk was really designed to show you that regulatory bodies do have the ability to respond quickly, even though it may appear that sometimes it takes longer than it should. But the real role of a regulatory body is to protect public health. And so these two mechanisms go a long way in doing that. I'm going to try to keep a similar theme for the second part of the talk, which is keeping up with technology. And you'll see these two little fellows here appearing as a continuing theme. You'll notice one's getting closer to the other, which is good. So here's the good news. Advances in technology have tremendous potential to address significant individual and public health needs. For example, personalized medicine is making great strides. There are improved detection techniques for infectious agents. There are diseases and medical conditions for which there are no tests, and platforms are being developed and systems are being developed to detect these. There are promising technologies available for earlier detection of cancer, which have greater accuracy to save patients from procedures that are very invasive and potentially extremely risky, such as surgeries, unnecessary surgeries. There's also greater reliance on complex algorithms and artificial intelligence. There is a tremendous increase in the use of computer systems and in algorithms that not only run the test but also learn as they go through artificial intelligence. I can tell you from personal experience that the group that I led at the FDA consisted of, at the time, one person who did software. After I left, and she actually retired as well, FDA hired 3 software engineers. These are people who were trained as software people just because there is such a heavy reliance for diagnostics on software. All right, so that was what the good news is. Here are the challenges. How does a regulatory body determine safety and effectiveness where there isn't any precedent? You have to study, especially for new technologies. Second, how does it do this both efficiently and effectively? Efficiently, that it is doing it in the shortest amount of time possible and not to hold things up because of their lack of knowledge, but also to do it in such a way that it works. How does it determine if the benefits outweigh the risks? And this is also a continuing theme. The regulator is going to make sure that if risks are present, and they always are, then what will the benefits to be gained from that test or procedure outweigh those risks, which will give it public health or give it clinical benefit. So how do we begin? First, develop scientifically sound means of assessment and determine an appropriate, in all capitals, level of safety and effectiveness. And I'll describe what I mean by that in just a few minutes. So, regulatory bodies need not or should not act in isolation. Input from stakeholders is critical. This involves public meetings, it involves conversations, it involves discussions, it involves publications. And to get input from subject experts, from laboratory professionals, the medical community, public health agencies, patient advocacy groups, and civil society. All of these people have a vested interest in the products that come out of a regulatory agency, and those agencies need to hear from all stakeholders, and that would involve everyone in this room, such that the regulatory agency is informed about what the needs are for each of these groups. So, how to continue? First, encourage manufacturers and developers to meet with regulators to demonstrate the technology. Regulators can't work in a vacuum. One of the most exciting parts of my job was to hear about the new technology, especially technology that I was not aware of, and to see demonstrations, to get my hands on the new technology and to see what it was, what the potential is, and then offer recommendations on how to assure safety and effectiveness. Regulators should be given the opportunity to attend, not only attend, but to participate and be active participants in meetings to learn what's on the horizon. Many meetings have manufacturers showing their new products. The regulators need to be there and go from booth to booth and see what the new technology is to allow the regulator to have a better idea of issues that might need to be addressed. Also, regulators should be proactive, not to sit back and wait for people to come to them, but to go out to others to make sure that they have the appropriate background to make intelligent and innovative decisions. This also involves holding public meetings when there are critical decisions to be made. It's very important to maintain a level playing field, as I call it, such that fairness is essential. There is no room for bias or even the appearance of bias toward one company or toward one technology. The mind of the regulator needs to be open to all possibilities and to provide feedback. Also consider alternative approaches. An agency may have a particular way of demonstrating safety and effectiveness, but the manufacturer may come up with an alternative way. As long as that can be scientifically and statistically validated or supported, justified, then the agency should be willing to accept a proposal that is so-called outside the box. That was actually the fun part of the job, also to hear about the ideas the manufacturers have in showing whether their products actually work or not. So let me give you an example, actually a couple of examples of how FDA handled and is handling innovation. The first example I want to give is for HIV self-testing. So, test performance is usually a straight line. Demonstrating safety and effectiveness through test performance is typically a straight line. And what that means is that a regulatory agency will say that for a given test, it has to be 98% sensitive and 98% specific. Okay? And sometimes those numbers can be justified, sometimes they just sound good. However, I want to introduce the concept of the risk analysis and to take into account public and individual health implications. So, if you have a test that performs at a particular level and it's below what you expect that to be, should you be able to accept that? And the way you can answer that question is by conducting a risk analysis to see how many false positives, how many false negatives you're going to get as a result of using a test like this, while at the same time meeting a critical public health need, such that you can have a bit of an indirect way of demonstrating safety, not indirect, but a way of demonstrating safety and effectiveness which you hadn't really thought was appropriate. So, the example I'm showing you here is for the OraQuick in-home HIV test kit. This was the first over-the-counter test, first self-test approved in the US. And the specificity of the test was, as presented to the FDA through data, the lower bound of the 95% confidence interval was 99.9%. Whereas the original recommended performance to FDA through advisory committee meetings, and that's also important to have the outside advice to a regulatory body, the recommended level was 95%. As the lower bound of that 95% confidence interval. But look at the sensitivity. The sensitivity was recommended to be 95%. Also, it turned out to be 86.6%. This is an HIV test, okay? So there were two options. One is for the FDA to say, go back, just do some more studies, change your test, and see if you can give us a test that's actually going to be performing at 95%. Or to take one step further, and I had some brilliant colleagues who I worked with who developed a risk model. And the risk model was designed to show how many people would learn their HIV status in the first year of use for a test that performed at this level who wouldn't otherwise be tested. Okay, these people just don't know their status, they hadn't been tested before. By providing this test, they would now learn their status. And also to understand how many HIV transmissions would be averted as a result of using this test. Public health benefit to the OraQuick test in terms of the increased number of HIV infections that were newly identified in the first year, and the net transmissions averted. For the transmissions averted, the number was approximately 4,000 HIV transmissions that would have been averted if this test were available. Just to give you some perspective, the number of true positives to false negatives was 6 to 1. So 6 true positives for every false negative result as a result of using a test that has 86.6%. This is an oral fluid test, by the way, which I believe has actually been accepted by Anvisa. So six true positives to 1 false negative. The question is that good enough? Are you willing to accept the false negative for every 6 true positives you identify? There is no right or wrong answer here. But we did take this to the advisory committee, and the committee recommended that the OraQuick test be approved because of the benefit. Now, I mentioned earlier that there also is going to be risk associated with this, and you can certainly see that, and that remains in the form of increased numbers of false negative results, and the response to that is to try to mitigate that. You have to put some responsibility on the part of the user. So this was done through labeling. That's typically the way that risk is mitigated for an IVD. And there were 4 messages that came across. One was, if you had a positive result, it doesn't necessarily mean you are infected. You need to have that followed up with a confirmatory test. Standard stuff. The second part is, if you have a negative result, it doesn't necessarily mean you're negative. And that usually draws some laughter from people because, well, if you're positive, you're not necessarily positive. If you're negative, you're not necessarily negative, so what good is it? I'll get to that in a second. But with a negative result, the window period for detection of antibodies to HIV for tests like this is about 3 months. So if you're testing within that 3-month period, it's not going to be as accurate. 2 other messages: don't use this test as a basis for engaging in high-risk behavior. Okay, don't take the test and say, "Ooh, I'm negative, you're negative. Great, let's increase the risk." And the fourth one was, if you are engaging in high-risk behavior, test on a regular basis. So again, putting responsibility on the part of the user as a way to, and through messaging, try to reduce the risk. One more example. This is called down classification, and for that I'm going to have to describe to you what classification means. There are 3 risk classes for medical devices in the US. Typically, regulatory bodies do have a risk classification system. It varies between 3 and 4 levels. FDA uses 3. First class is the class with the least risk, and for this, general controls over the product are sufficient to assure safety and effectiveness. For class 2, which is the middle one, also known by some, if you know any regulatory jargon, in the US it's referred to as a 510K. You may have heard of that. So for this, general controls alone are not sufficient. General controls, by the way, are such things as manufacturing, quality system, and that sort of thing, and reporting of adverse events. But general controls in and of themselves are not sufficient, but special controls would be needed to assure the safety and effectiveness, and pre-market notification or submission of this 510K document would be sufficient. So there would be less documentation, no pre-market inspection, lower fees, and comparison to what's called a predicate device. This is all as compared to the riskiest of the devices, which are class 3. For this, special controls aren't sufficient, so they have to go through pre-market approval. Again, reserved for the highest risk of the medical devices. It involves a pre-approval inspection, extensive documentation, longer review times, and higher fees. There's your background. FDA has the ability to both up-classify, in other words, to make something more stringent if they know that there's more risk associated with it, or to down-classify, which means that they've decided that things aren't as risky as we thought they were before. Or there have been some events that have taken place which show that the test is not as risky as originally thought. I'm going to show you a couple of cases just on 2 slides, this and the next. So the first, and these may come across as a bit surprising. The first down classification from class 3, the most stringent, to class 2, the middle one, was for tuberculosis tests. And I'm sure everyone in this room would agree that tuberculosis test is a pretty significant risk device. But FDA's experience was that for nucleic acid-based IVDs, for detection of TB, and for detection of genetic mutations, that the risk, after reevaluation, the risk was such that it could be down-classified, making the process easier, the regulatory process easier, for tests such as this. This took place about 3.5 years ago or so. This does not affect serological tests, by the way. So serological screens for TB still remain at class 3, but the nucleic acid-based tests were down classified to class 2. And then another one which is actually near and dear to my own heart, since I was regulating HIV diagnostics, about 3 weeks ago, there was a meeting at the FDA, a panel meeting. This was an advisory committee meeting, which actually the first one didn't take place, but is about to because we were snowed out. But the discussion was for reclassification of HIV point-of-care and laboratory-based serological and NAT test, nucleic acid diagnostic tests, from 3 to 2, and the same for HCV, for hepatitis C. Now the general rule, not general rule, the convention was that you can do it for HIV, you can do it for anything. HIV was always the benchmark for high risk. In this case, FDA is now saying that they, based on experience and based on technology, that they feel comfortable down-classifying this from the highest risk device to a middle risk device. The HCV meeting actually did take place, and there was there was consensus unanimously from the committee to move ahead with down-classifying HCV tests. I will be very surprised if the same does not happen to HIV. So this is all meant to show you that FDA, as an example, but regulatory bodies do have the ability to make products move through the system faster and more efficiently. So, some concluding thoughts, just to summarize what I said. Regulatory systems are necessary to ensure that products are safe and effective for their intended use. And the system, but at the same time, the systems need to be flexible so that technology can be developed and available to support individual and public health. Flexibility takes into account whether the benefits outweigh the risks, and flexibility is critical to allow those products to get out the door. Again, contrary to what some think, the regulatory agencies actually want to get good products on the market. At the same time, they're trying to prevent bad products from entering the market. And lastly, it's critical that regulatory bodies are partners in product development and not act on their own, so that appropriate, that should be capitalized, regulatory parameters can be set to ensure that critical technology does in fact come to clinical use, but it's such that that clinical technology is indeed safe and effective. So with that, where we finally caught up, I hope, we overgot it.